أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Today we are going to discuss lecture number 6. This is about the history of microbiology. Actually, there are 37 important names regarding historic work done in microbiology. First name is Guillermo for Castro. He gave the theory about the epidemic diseases, that epidemic diseases are contagious and disseminated by minute particles carried over long distance. Number 2, second name, very important regarding the history of microbiology, is Zacharias Janssen and Hans Janssen. He developed first compound microscope and illumination system. So first compound microscope developed by Zacharias Janssen and Hans Janssen. Number third name is Robert Hooke. Robert Hooke, he was the first man to observe the cell. Number 4, Carl Linnaeus developed hierarchical classification of all organisms and binomial nomenclature system. Fifth name, very important, is Lazzaro Spallanzani. He did first experiment refusing the theory. Of spontaneous generation and the first growth of bacteria in the culture. Number 6, we are just going through all these names. Number 6, Edward Jenner. Application of cowpox virus for vaccination against smallpox. This was the work done by the Edward Jenner. Number 7, Agostino Bassi. He was the first man who identified white muscardine fungus from the silkworms. Number 8, Anton de Barre. He is considered as the father of plant pathology. Number 9, Louis Pasteur. This was the man who refuses the doctrine of spontaneous generation. He patented the process to fight the diseases of wine in 1865. The method became known as pasteurization, and this Louis Pasteur is considered as the father of modern microbiology. Very important man. Number 10, Joseph Lister. He is considered as the pioneer in antiseptic surgery. Number 11 is Oscar Briefeld. He developed pure culture technique for isolation and culturing of the fungus. Number 12 is the Robert Koch. He is also well-known person regarding microbiology. He was the supporter of the germ theory of disease. Koch's discovery of theory of causative agent of anthrax led to the formation of a generic set of postulates which can be used in the dissemination of the cause of most infectious diseases. Next, very important man, number 13, is considered as Hesse. He used agar as a solidifying agent for culture media, and we all know that agar is not providing the nutrition to the bacteria. It is only solidifying agent for culture media. So Hesse was the man who used agar for this purpose, for solidifying agent for culture media. Next is number 14, Charles Chamberlain. He developed Chamberlain pasture. Procylane ultra filter. Number 15 is Hans Christian Gram. Hans Christian Gram developed Gram's staining technique. Very important man. Number 16 important man regarding microbiology is A.B. Frank. He discovered mycorrhizal fungal. Number 17, Anton van Leeuwenhoek. He is considered as the father of microbiology. Invention of simple microscope is related with this man. Next, 18, Richard J. Petri. Richard J. Petri developed Petri dish. Very important apparatus used in microbiology. Next is 19, Emil von Behring. He developed diphtheria antitoxin. Diphtheria antitoxin was developed by this man. Ivanovsky discovered tobacco mosaic virus, TMV. So tobacco mosaic virus was discovered by Ivanovsky. And 21, Paul Ehrlich, side chain theory of antibody production was given by this man. Number 22, Fleur. Fleur discovery of foot and mouth disease. Actual word is not foot, this is foot and mouth disease. So this Fleur is related with the discovery of foot and mouth disease. So mistake has been corrected. Next man, very important regarding microbiology history, is Stuart. He discovered bacteriophage. Stuart discovered bacteriophage. 24, Griffith. Griffith is the man who discovered transformation in bacteria. This is a process very important regarding bacteria. So Griffith is the man who discovered transformation in bacteria. Number 25 is Alexander Fleming. Alexander Fleming discovery of penicillin. This penicillin was the first antibiotic. 26, Ernst Ruska. He invented electron microscope. 27, Stanley. Purification and crystallization of tobacco mosaic virus was done by this Stanley. We are just going through all these names. These 37 names should be remembered by each and every microbiologist. 28, Barbara McClintock. He development of the concept of transposal element or transposons. Beadle and Tatum, one gene, one enzyme hypothesis was given by these two persons. S.A. Waxman developed streptomycin. So Waxman developed streptomycin. Remember this. Lederberg-Tatum discovery of bacterial conjugation was done by this Lederberg plus Tatum. Lederberg plus Zinder. Discovery of transduction, transfer of genetic information between bacteria by the virus is known as transduction. So this discovery of transduction is related with Lederberg plus Zinder. Diner is the man who discovered the virions. Kohler and Milstein development of monoclonal antibodies was done by these two persons. Vose, first person to identify Archaebacteria. And number 36, T. Brock. This man discovered the polymerase, TAA polymerase. Prociner discovered prions. So prions were discovered by Prociner. This was all about the historic work done by these 37 scientists, microbiologists. We must remember all these. Thank you very much. We'll come with the next lecture inshallah very soon.